Forget everything you've been told about basic sleep hygiene. You know, the standard advice about avoiding blue light or lowering the thermostat. I mean, it's fine, but it barely scratches the surface of human biology. Today, in this explainer, we're going to completely deconstruct human consciousness, recovery, and longevity, and we're doing it from first principles. We are diving into a unified biological framework to explore exactly how our brains transition between fiery, waking arousal and those restorative depths of sleep, right down to the cellular, molecular, and electrophysiological levels. It's going to be a wild ride.
So here's our roadmap for today. First, we'll look at the architecture of sleep. Then, the VLPO multi-system integrator. Third, serotonin's dual arousal engine. Fourth, the orexin stabilization circuit. Fifth, respiration and autonomic control. And finally, number 6, optimizing deep slow-wave sleep. Let's get right into it.
Section 1. The architecture of sleep. All right, to really master our nervous system, we first need to understand the big, overarching systems governing our biological rhythms. Because sleep, it isn't just a switch that suddenly flips when you're tired. It's actually the precise intersection of two really dynamic forces. We call this the two-process model. Process C is your circadian rhythm. Think of it as a biological clock, mostly driven by the suprachiasmatic nucleus, that aligns your physiological timing with the actual rotation of the Earth. Pretty cool, right? Then you have process S. This is your homeostatic sleep pressure. As your neurons are firing away throughout the day, spending metabolic currency, byproducts are constantly accumulating. Specifically, adenosine builds up in the brain. So it's this interaction between that rising metabolic pressure and your circadian clock that ultimately dictates your biological need for deep recovery.
So being awake versus being asleep, it literally represents this epic biochemical tug of war. Wakefulness is this fiery, high-energy arousal state, and it's driven by heavy-hitting neurotransmitters like serotonin and orexin. On the flip side, the restorative depths of non-REM and REM sleep are totally dominated by inhibitory signals, specifically GABA and glaninergic signaling. The key takeaway here is that when you fall asleep, your arousal system doesn't just quietly fade out into the background. No, it is actively conquered and suppressed by the brain's sleep networks.
Which brings us to Section 2, the VLPO multi-system integrator.
Okay, so where exactly does this active suppression of wakefulness actually come from? Well, for decades, scientists looked at the ventrolateral preoptic nucleus, or VLPO, which is buried deep in the anterior hypothalamus. And they just thought of it as the brain's sleep center, like a simple on and off switch. But that view is fundamentally outdated now. The VLPO is actually this incredibly sophisticated multi-system integrator. It takes in an immense amount of biological data. We're talking core body temperature, metabolic status, inflammatory markers, homeostatic sleep drive, the whole shebang. And it continuously calculates whether it's biologically safe and appropriate for your body to initiate sleep. So how does the VLPO actually know you're tired, or say, well fed? It does this through very precise converging molecular signals. For instance, wake-dependent energy consumption creates adenosine, which then binds to A2A receptors to promote sleep. But survival requires a lot more than just being tired, right? Interestingly, energy availability from circulating glucose actually excites sleep-promoting neurons through these specialized potassium channels called KATP channels. Even leptin, which is your satiety hormone, feeds into this system. Basically, the VLPO biases your microcircuits toward NREM sleep only when it confirms your nutritional and energy state is secure enough to power down for the night. Moving on to Section 3, serotonin's dual arousal engine. All right, prepare to have a major misconception completely shattered. 14. That is the number of distinct, known serotonin receptor subtypes in the human body. Culturally, we tend to think of serotonin exclusively as this calming, happy molecule, right? Well, that couldn't be further from the truth. In the context of sleep and arousal, serotonin, which fires primarily from the dorsal raphe nucleus, is actually a massive, powerful arousal engine. It promotes intense wakefulness and actively inhibits REM sleep. So we're not just looking at one simple signal here. It's a vastly complex and intricate system. Serotonin's real complexity lies in how it interacts with different cellular machinery. Take 5-HT1 receptors, for example. These are coupled with GI and GO proteins, and they decrease cellular secondary messengers like CAMP. Basically, they act to quiet or inhibit downstream neurons. But then you have the 5-H2 and 5-H3 families. 5-H2 receptors increase intracellular IP3, which triggers calcium release, while 5-H3 acts as a literal ion channel. Both of these are highly excitatory. By deploying serotonin across these varied receptor families, your brain can intricately fine-tune your entire nervous system, keeping you alert, reactive, and fully conscious.
Section 4. The orexin stabilization circuit.
Now, with so much powerful excitatory neurochemistry flying around, you might be wondering, how does our consciousness actually remain stable? The answer is found in one of the most elegant molecular feedback loops in all of human biology. Let's break it down. Step 1, orexin neurons, which are driving wakefulness, excite the dorsal raffinucleus. Step 2, this triggers a massive release of serotonin. Step 3, that same serotonin circles right back and hits specific 5-HT1A inhibitory receptors on the very orexin neurons that started the whole cycle. This opens GIRK channels with a staggering precision of 33.8 picosiemens. That is an unimaginably tiny exact electrical conductance. And finally, step 4, the orexin neurons are hyperpolarized. They are essentially pumping the brakes on their own activity, so your arousal system doesn't just spiral completely out of control.
And we know exactly what happens when this precise causal loop breaks down. When researchers engineer mice that lack these specific 5-HT1A receptors on their orexin neurons, they're known as OX5-HT1A RKO mice, their sleep architecture just completely collapses. Without that serotonergic bake we just talked about, they suffer from highly fragmented wakefulness and completely disrupted REM sleep homeostasis. They literally cannot maintain stable consciousness, and they show these abnormal, premature transitions between waking and sleeping states. It's a biological mess without it.
Section 5, respiration and autonomic control.
So as we descend into deep sleep, our conscious control over our body just vanishes, right? So who's driving the autonomic nervous system? Once again, serotonin plays a pivotal, literally life-sustaining role. Medullary serotonin neurons, which are located right down in your brainstem, are absolutely critical for sensing carbon dioxide in your blood and maintaining eupneic, or normal, respiratory rhythms. Think about it. If your airway becomes compromised while you sleep, it is this specific network that detects those rising CO2 levels and triggers the autonomic arousal you need to just take a breath. This forms a direct, mechanistic link between serotonin levels and conditions like sleep apnea and sleep-disordered breathing. And finally, Section 6, optimizing deep, slow-wave sleep. Okay, so we've mapped the pathways from circadian timing all the way down to precise channel conductance. But how do we actually leverage this framework to maximize our own recovery? Well, for decades, standard sleep medicine really relied on traditional hypnotics, like benzodiazepines. But the trade-off there is severe. They artificially knock you out by broadly inhibiting the system via GABA. Ironically, this actually reduces your deepest, most restorative phases of slow-wave and REM sleep. Now compare that with targeted 5-HT2A receptor antagonists. By specifically targeting the serotonin system, these compounds don't just sedate you, they actually normalize your sleep architecture and significantly boost the duration of restorative slow-wave sleep. It's a real game changer. To understand exactly why this works, let's tie it all back to our first principles. Remember how we said that 5-HT2 receptors are those excitatory arousal engines? Well, selective 5-HT2A antagonism essentially unplugs the excitatory wake-promoting effects of serotonin right at the receptor level. It safely disables the brain's internal alarm clock. By doing this, it tips that systemic biological tug-of-war entirely away from arousal, allowing the VLPO's inhibitory signals to just fully saturate the brain, which drives you into deep, uninterrupted, physically repairing NREM sleep. So, we've traversed the cellular architecture of sleep, we've debunked the whole serotonin myth, and exposed the neurocircuitry that literally stabilizes your reality. True sleep optimization, it's not just about going out and buying a better mattress, it is about aligning your daily metabolic inputs, your light exposure, and your physiological stress with these hardwired, ancient neurobiological pathways. So I'm going to leave you with this one thought. Armed with the precise molecular mechanics of your own consciousness, how will you leverage this biological framework to master your nervous system, enhance your recovery, and secure your long-term health span? It's a lot to think about, right? Thank you so much for joining me for this explainer. Keep optimizing, and I'll see you in the next one.